This section explains why every generation of biofuel is a single move along one axis, how hard the carbon is to reach. First generation fuel taxes the plant's own hoard, seed oil, cane sugar, the storage organs it already concentrated for you, which is why it collides with the food supply. The frontier is the structural carbon, cellulose and lignin, the scaffolding a plant evolved specifically to resist being broken down. That recalcitrance is the whole problem, and synthetic biology is the deconstruction key. Engineered enzymes and organisms that crack lignocellulose into fermentable sugar, then push further into designer molecules and algae that never touch arable land. The chapter opens the book by making biofuels its flagship case, the industrial payoff gated on unlocking the carbon nobody else can. This section explains the single structural move behind every biofuel. Every biofuel is stored sunlight. The whole field reduces to one move. Drop an engineered organism in as the catalyst that turns light fixed carbon into something combustible. The spine of the chapter is the generational ladder. Sugars and oils from plant storage organs come first, recalcitrant, lignocellulose, second, algae and direct artificial photosynthesis, third. Read these three rungs as one monotone trade-off rather than as three separate technologies. Each rung climbs away from food and land competition by demanding a harder biological deconstruction, and synthetic biology's task is precisely to pay that rising catalytic cost. Predict the next instance yourself. Any candidate fuel sits somewhere on an axis running between ease of conversion at one end and freedom from the food supply at the other, and the engineering is whatever closes the gap. This section explains the L1 structure of bioremediation. Pollution meets metabolism, and the chapter asks a single question. Can you engineer a microbe whose ordinary chemistry happens to eat your poison? The thesis is the honest no. Yet underneath the hope, two verbs exhaust the space. The first is to degrade the pollutant into something harmless. The second applies when it cannot be degraded, as with metal ions, arsenic, radionuclides, and minerals, which have no bonds a cell can profit from breaking. There, the microbe can only concentrate and precipitate the pollutant, relocating it rather than destroying it, relocating it rather than destroying it. The correction that carries the chapter is this. Aromatics resist you the way lignin keeps wood from rotting, and worse, they dissolve the very membrane that maintains the microbe's viability. The cleaner is itself the target. Cleanup that self-poisons must lean on physicochemistry too. This section explains why bioremediation is two operations, not one. Bioremediation is metabolism recruited as reversal, life set to undo what industry dispersed. The thesis of the chapter is that this splits cleanly into two inverse operations, never one. Organic pollutants. You dismantle. You break the aromatic ring, and the tell that this is hard is wood whose lignin backbone is aromatic, which is precisely why it refuses to rot. Toxic minerals you cannot dismantle, only relocate. Microbes concentrate them and precipitate them as insolubles, the exact inverse of leaching metals out of ore. Both presuppose a third operation, detection. You sense the pollutant before acting. The surprise that carries the chapter is the death of the naive hope that one engineered microbe could simply eat any poison. It couldn't. The ring resists. This section explains the first primitive of the book's industrial arc. a biomaterial grows rather than gets synthesized. You cultivate the material rather than refining it from petroleum. The one primitive this opening chapter lands is that manufacture becomes metabolism. The move that does the work is a basis. Rather than engineer a bespoke pathway for each useful chemical, synthetic biology fixes about 12 sugar-derived building blocks and reaches the entire space of materials as compositions over that small set. A limited module library upstream, another downstream, no full pathway rebuilt per product. Factor the materials, economy through a spanning set, and the combinatorics collapse. That is the whole bet. The following sections cash out. This section explains how Chapter 3 works. Chapter 3 is the book's assessment template applied to one class of output, biomaterials, the substances biology makes, reframed from found to engineered. The generative move is a fixed 4-axis rubric. The 4 axes are technical potential, economic potential, environmental impact, and social and ethical stakes. That same rubric runs identically over each material family: fine chemicals, bulk chemicals, bioplastics, and cellulosomes. Read one family's 4 scores and you can predict the next. The chapter simply is that repeated evaluation itself, the same rubric run again rather than a survey of substances. The reframe that carries the whole chapter is this: synthetic biology turns a material into a designed molecule, building blocks made from sugar, the DOE12 of Figure 3.1. So what a biomaterial is collapses into what it does. This section explains 2 ways to do synthetic biology that leave the genome behind entirely. Mainstream synthetic biology hacks the existing genome from the top down, editing life's code in place. This chapter catalogs the 2 ways to leave the code entirely. The first way is to build below it. This is protocells, life-like droplets, self-assembled from raw chemistry. They are mobile, they metabolize, they move toward chemical signals, yet they carry no DNA. The second way is to build beside it. This is xenobiology, swapping the alphabet itself. Xenonucleic acids, mirror chirality, expanded codons. The unifying move is orthogonality, a component whose changes cannot propagate into the surrounding system. Here is the surprise that makes the whole thing go through. The very non-participation that makes these approaches other is exactly what makes them safe. No shared DNA, no shared alphabet, and therefore no horizontal gene transfer. This is a genetic firewall against contaminating the wild. This section explains how the 4th body of chapters breaks from everything before it. Every prior chapter reprogrammed existing cells, top-down hacks on DNA that stays fluent in the natural genetic language. This chapter breaks that frame twice. Protocells build life's minimum upward from bare chemistry. They are self-assembling oil droplets that move, interact, and shed skins with no DNA at all. Xenobiology rewrites the substrate itself, using xenonucleic acids and non-canonical amino acids, so the organism speaks a chemistry nothing natural can read. Both make incompatibility itself the design object. Orthogonality is the deliverable, the product itself, rather than a safeguard bolted on afterward. Re-engineering the cell buys function. Re-engineering the chemistry buys a genetic firewall, an isolated enclave that cannot exchange information with the living world. This section explains what governs synthetic biology and where its rules come from. No law was ever written for synthetic biology. It is governed by inheritance. The recombinant DNA regime of the 1970s retrofitted onto a field that did not exist when those rules were drafted. That is the chapter's single move, and the book's applied argument comes to rest on it. Everything demonstrated earlier is bounded by borrowed containment. The three jurisdictions, the United States, Europe, and China, are three instantiations of the same retrofit. Read comparatively. The surprise that updates your model sits in the enforcement gap. The most followed instrument, the NIH guidelines, carries no binding authority. It governs by funding contingency and de facto consensus, not by statute. Coverage comes by adaptation. Compliance runs on dependence, not law.